Hello again. This is the end of Chapter 3. We'll be talking about the organelles that we find inside of the cell in this video. So we'll be talking about the cytoskeleton and then all of the other organelles and talking about both their structure and their functions. Starting with the cytoskeleton, the whole idea of the cytoskeleton is that it gives the cell shape and support and is in some cases kind of like a railway system or a zip line system and allows for the transportation of materials within the cell in an organized way. We have three different types of cytoskeleton. We have microfilaments, intermediate filaments, and microtubules. And those three things are differentiated based on size. Microfilaments are the smallest. They're 6 nanometers thick. If we have filaments that are between 8 and 10, we call those intermediate, and then the thickest are called microtubules. Microtubules are actually hollow, which is kind of interesting. We see these in different places and they do different functions, but generally speaking, they give the cell structure and support. Moving on to talking about organelles, we have two main categories. We have membranous or membrane-bound organelles. These are things that have their own lipid bilayer. And then we have things that are not membrane-bound. It's probably a good idea to remember which ones have membranes and which ones don't. Could come up on your exam and will help you to remember what their functions are. So arguably the most important organelle is the nucleus. The nucleus is the largest thing that we find in the cell in most cases. Most cells have one nucleus. There are some that don't have any nuclei and some that have many nuclei, but typically a cell has one nucleus. The nucleus has a double membrane. So we have a double membrane around the nucleus, and that double membrane has a lot of pores in it, which are made out of rings of proteins. We can see them here in this micrograph. Those proteins hold the two membranes together and regulate what's going into and what's coming out of the nucleus. This nuclear membrane or nuclear envelope is supported by something called the nuclear lamina, which is a web of filaments like we just talked about, the microtubules and intermediate filaments. And those things regulate the things going on inside that nucleus. Inside of the nucleus, we have chromatin, which is the DNA. The DNA stays in the nucleus unless the DNA is replicating. And we also have the nucleoli, which is where ribosomes get produced and then sent out into the cytoplasm. And we'll be talking more about ribosomes in just a couple minutes. Moving on to the endoplasmic reticulum. The endoplasmic reticulum is one of the membrane-bound organelles. We have two varieties of endoplasmic reticulum. We have rough endoplasmic reticulum and smooth endoplasmic reticulum. We'll see on the next slide. The difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum is the presence of ribosomes. So if endoplasmic reticulum has ribosomes stuck on its external surface, we call it rough endoplasmic reticulum. If it does not have ribosomes, it is smooth endoplasmic reticulum. A lot of times we abbreviate endoplasmic reticulum as ER. So rough endoplasmic reticulum you may see as RER, and smooth endoplasmic reticulum you'll commonly see as SER. Smooth endoplasmic reticulum, like we just said, it lacks ribosomes. This is the place where steroids and lipids, remember steroids like testosterone, estrogen, cholesterol, and other lipids, this is where they get made. The smooth ER is also where the reactions happen that deactivate alcohol and basically digest the alcohol. We also store calcium here. Rough ER and smooth ER are connected. The thing that distinguishes them is the presence of the ribosomes. On to ribosomes. Ribosomes are made of protein and RNA. They're a complex of these two things that might be important on an exam. We find ribosomes in the nucleoli, which is where they're synthesized, and also in the cytosol or cytoplasm. They can be attached to the endoplasmic reticulum, making rough endoplasmic reticulum. Sometimes they just float freely in that cytoplasm or cytosol, though. The job of ribosomes is to read the genetic code that is carried by messenger RNA, and then to assemble amino acids in the proper order to make proteins. Once those proteins are made, the Golgi complex receives the new proteins that come from the rough endoplasmic reticulum and the ribosomes. It essentially finishes those proteins. It can add different elements to the proteins, and it can package them so they're ready to transport within the cell or out of the cell. So the Golgi complex puts the finishing touches on proteins after the ribosomes have assembled them. Lysosomes and peroxisomes are things that have some similarities. So I put them side by side here so that you guys are able to compare and contrast easily. They are both membrane bound, and they both do forms of digestions. So lysosomes are full of enzymes surrounded by a membrane, and they break down dead or dying cell parts. Okay, so they can digest proteins, nucleic acids, carbohydrates, lipids, all sorts of things. They have a variety of enzymes that are very good at digesting dead and dying cell parts. We have some diseases that occur when lysosomes don't have their proper membrane. Imagine we have these digestive enzymes just floating around in cells. The cells would be very unhealthy, right? So lysosomes are really important. Those lysosomes having the proper membrane around them are really important in keeping your cells healthy. Lysosomes also function in something called autophagy. Autophagy is what happens when we need to digest an entire organelle, or autolysis when we need to let the whole cell be digested and the components recycled. Peroxisomes in some ways are similar to lysosomes, but they do a specific kind of digestion that is done by hydrogen peroxide. So hydrogen peroxide is used to neutralize free radicals. We talked about those earlier. They're things with strange or weird numbers of electrons in their electron configurations, and we need to get rid of them before they can damage cells. Peroxisomes are the organelles that do that. All cells have some peroxisomes, but cells that are in the liver and in the kidney, where we are dealing with waste and toxic compounds, have really high levels of peroxisomes. And here's a picture of lysosomes and peroxisomes next to each other. They look very similar. They're membrane-bound things that have enzymes in them that break things down. Proteasomes are specific things that can digest proteins. Okay, so proteasomes are also involved in digestions like peroxisomes and lysosomes, but they only break down proteins. Mitochondria are super important organelles. Their specialty is producing ATP. Okay, so we've talked about ATP getting produced via glycolysis, and then the Krebs cycle, and then oxidative phosphorylation, or the electron transport chain. The Krebs cycle and electron transport chain happen in mitochondria. So most of our ATP is getting produced in mitochondria. You guys may have heard in middle school and high school, mitochondria are the powerhouse of the cells. So if that helps you remember what's going on here, that is a useful memory trick. Mitochondria actually have a double membrane, and they have an external and an internal space. And finishing up with centrioles. Centrioles are short pieces of microtubules that are involved in cell division. So they have this kind of unique star-shaped arrangement, and when we look at a cross-section, like straight across this centriole, this is what we see, with the different microtubules arranged in a very specific way. These are important in cell division. That's an important thing for you guys to remember. Also important in attaching cilia and flagella, those two things that are projections from the cell surface that can allow cells to move and serve as receptors.